RNA World and Replication of DNA. RNA was the first genetic material that was involved in essential life processes like metabolism, translation, and splicing. But since the RNA is a catalyst, it is reactive and unstable. Hence, the double-stranded DNA was evolved from RNA in order to resist changes in the organism. RNA catalysis takes place in the biochemical reactions in living systems instead of using protein enzymes. The catalytic RNA or RNA enzymes are known as ribozymes. Replication. DNA carries genetic information. It has two functions: heterocatalytic and autocatalytic function. Heterocatalytic function. In this function, the DNA directs the synthesis of chemical molecules, such as proteins. Autocatalytic function. In this function, DNA directs the synthesis of DNA itself, that is, replication. DNA replication. It is an essential biological process that occurs in all living organisms to make more copies of their DNA. Since DNA is the basis for inheritance. Rules of DNA replication. DNA replicates in a semi-conservative mode. The replication may proceed in either one or both the direction from a given point. The unique point from where the replication starts is called the origin of replication. Strong replication occurs when the addition of nucleotide monomers happen in the 5-3 direction. Replication occurs in the short discontinuous pulses due to which we get short fragments. These short fragments are joined to form a complete strand. A short segment of RNA primer initiates the action of DNA polymerase, which adds the nucleotides to the grow of a DNA strand. The experimental proof: semi-conservative model of DNA replication. Matthew Meselson and Franklin Stahl proved that the DNA undergoes a semi-conservative type of replication. They performed an experiment in which they grew E. coli in a medium with 15NH4Cl such that it is the only nitrogen source. 15N is the heavy isotope of nitrogen. After the first stage replication, DNA was extracted and centrifuged. Heavy DNA molecules were distinguished from the regular DNA molecules and then transferred again into a medium with 14NH4Cl. The samples showed that the DNA collected after the transfer of 20 minutes had a hybrid, that is, the first generation, and the DNA collected after 40 minutes had equal amounts of hybrid DNA and light DNA, that is, the second generation. This experiment proved the semi-conservative mode of replication of DNA.